Hi everyone. Now we are going to discuss a topic called electrophoresis. Yes, electrophoresis. In this topic also, we are mainly dealing with principles and applications of electrophoresis. So this is an electrophoresis diagram that it is going to show, and these are all the different bands of the molecules that you are going to separate. Okay, so the separation of the molecules we had seen in a technique called as chromatography, and this electrophoresis is an advanced form of this chromatography. So you can go through the chromatography topic that already there in the other videos. Okay, let's come into the topic of electrophoresis principles and applications. So what is an electrophoresis? Electrophoresis is a process of moving charged molecules in solution by applying an electric field. Remember, in general also the molecules are moving from one position to the other, but because of an electric field the charged molecules are moving. Okay, so field across the mixture is going to be termed as electrophoresis. So electrophoresis is a process of moving charged molecules in a solution by applying an electric field across the mixture is being termed as electrophoresis. This electrophoresis technique was first invented by Mr. Tislius in the year 1937. In the year 1937. Why he did it? Because molecules in an electric field moves with a speed, yes, obviously, depending upon their charge, shape, size. Okay, so all these things are going to have a major impact in the mobility of these molecules. So electrophoresis has been extensively developed for molecular separations. What do you mean by molecular separations? That is your DNA, RNA, proteins, all those things can be separated very easily by using this electrophoresis. So then coming to the next, so this is a chamber of the electrophoresis. So if you observe, here is a loading of the sample, the dark blue in color. Okay, and here is a well. So there will be a well. Well does not mean of your water. This small nod like structure which we call it as a well, and in this well you are going to have your uh, putting your sample. And if you observe, here is a circuit connected to it of having the cathode and the anode parts, and after the power supply the molecules that we have placed the sample here, but here if you observe there are different bands. So these are all the molecules that are separated from the sample depending upon their size, shape, mobility. So all these things we are going to have the different bands. So lower molecular weight obviously comes faster, that means we will see at the bottom, and higher molecular weight some molecules are going to be at the top of the thing. So this is how you are going to have a separation of molecules from a mixture by applying the electrophoresis. Then coming to the purpose of carrying out electrophoresis. You may all get doubt why we have to use this electrophoresis. I told you already in the initial step that it is majorly used in the molecular separations, isn't it? Your DNA, RNA, all the things. So this electrophoresis is mainly used to determine the number, amount, and mobility of components in a sample or to separate them. So that's the main purpose, to determine the number, amount, and mobility of components in a given sample. Then to obtain information about the electrical double layers surrounding the particles. Every particle is going to have some sort of a layer surrounding them, and that layers which type of the charge is going to be present, everything we'll come to know by using electrophoresis. Not only that, as I told you, we can determine the molecular weight of proteins, and even in the DNA sequencing also we can use this electrophoresis. So these are the main purpose for which we are carrying out the electrophoresis. Then coming to the principle behind this electrophoresis, that's what I told you. We are majorly concerned about the principle and applications of electrophoresis. So coming to the principle, Any charged ion or a molecule migrates when placed in an electric field, isn't it? When placed in an electric field. The rate of migration depends upon its net charge. So, what is the charge of it? What is the size of it? What is the shape of it? See, the spherical shaped substances will move faster when compared to having a rectangle or a square shape, isn't it? So, the shape also plays an important role in the migration. Then the size. The heavier molecules will stay back and the lighter molecules will move faster. And the applied current. So how much of the current, that volts current you are applying, also is going to have the factor in the movement of the molecules. And this whole thing is going to be represented by a formula or equation called as V is equal to EQ by F. What is it? V is equal to EQ by F. So what is this V? What is this U, Q, and F? Let's see. V is the velocity of the migration of the molecules. How much fast it is going to move within this time we are going to term as velocity. And the E, that is electric field, that how much of the electric city that you are going to provide in volts per centimeter. And the Q indicates the net electrical charge on the molecule. Okay, and the F is going to denote the frictional coefficient. So this frictional coefficient, as I told you, depends upon the mass and the shape of the molecule. So if you're having the circular figure, you're going to have the less friction, and if you're having a more mass and the shape, obviously you're having the more frictional. So these are the things that we are going to have majorly working in the principle of electrophoresis. So V is equal to U EQ by F. Okay, now the movement of charged particles in an electric field is expressed in terms of electrophoretic mobility. Electrophoretic mobility, which is being denoted simply by mu. This mu is equal to Q by F. What is the Q by F? The Q is going to be the net electric charge and F is the frictional coefficient. Or simply we can take the velocity of the migration of molecule divided by the electric field that we are using in the volts and centimeters. But so this is all about the principle on which it is going to, the electroforces is going to work. For molecules with similar conformation, F varies with size but not with the shape. Remember, the frictional coefficient is going to varies with the size but not with the shape. Okay, so if you're taking the larger size sphere and the small size sphere, there the shape is the same, but the size is going to be different. So this is going to vary with the size but not with the shape. Thus, electrophoretic mobility, that is the mu of a molecule, is directly proportional to the charge density. Okay, so charge by mass ratio is going to be called as charge density. So your mobility is equal to or directly proportional to the charge density of the molecules. So this all about the principle of electrophoresis, which is very important in the exam point of view. Okay, then moving to the applications of electrophoresis. So we are supposed to learn about the applications of any instrument that we are using, isn't it? So coming to the applications, electrophoresis is employed in biochemical and in the clinical fields. That's the first thing. That means we can use it in the biochemical field as well as in the clinical fields also, in the DNA sequencing, all those things. And we can also use this in the study of protein mixtures. So what are the different types of the proteins that are present in a concerned sample? And then it is also being used in the antigen antibody reactions, isn't it? In the clinical diagnosis, we can use this electrophoresis. And then in fractioning of the protein, sometimes it's very essential to fraction the protein, so obviously we'll do that one. And then in analysis of lipoproteins, serum, cerebrospinal fluid, your urine, hemoglobin, peptides, nucleotides, and some similar ions, you're going to have the analysis process also. We can use this electrophoresis mainly in the diagnosis purposes. And then these electrophoresis is going to be combined with some sort of a radiography in some sort of the techniques to use. And in food industry also this electrophoresis is being used. And not only in that, in separation of some organic acids, alkaloids, carbohydrates, amino acids, alcohols, phenols, nucleic acid, insulins, we can use this electrophoresis. So these are the few applications where we are using the technique called electrophoresis. So in the biochemical and clinical fields, in the study of protein mixtures, and then in the antigen-antibody reactions, then coming to the fractioning of protein, and then in the analysis of certain biochemical samples, and then in combination we can use this in the field of radiography, and then in the field of food industry we are using, and in chemical field also we are using this in the separation of all chemicals, as both organic chemicals and the biochemicals also. Okay, so this is all about the applications of electrophoresis. And then coming to the different types of electrophoresis, we are having several types. Okay, free electrophoresis and zonal electrophoresis. The free electrophoresis are again of two types, that is microelectrophoresis and moving binary electrophoresis. And then coming to the zone electrophoresis, we are having majorly three types, that is paper electrophoresis, cellulose acetate electrophoresis, and then the next one is the gel electrophoresis. So we are not going to deal with all these types of electrophoresis, just we'll go in detail about this gel electrophoresis. And this gel electrophoresis is again being classified into three types, that is agarose gel electrophoresis, starch gel electrophoresis, and PAGE, simply called as PAGE, polyacrylamide gel electrophoresis. And these topics will be covered in the other parts. Okay, so in this part we have discussed about the electrophoresis basic principle, applications, and the types of electrophoresis. Thank you.